Okay, so we have covered glycogen breakdown and glycogen synthesis, and then kind of talked about this phosphorylation cascade that kind of helps regulate that process, particularly on the activation of glycogen breakdown side, where we're talking about pKa phosphorylating phosphorylase kinase, which phosphorylates phosphorylase, which again then breaks down glycogen. So again, this is a review slide from last time, but we're going to talk today about the last kind of player in this, which is protein phosphatase 1, and this is the main player for turning off not only this pathway, but other pathways. So like protein kinase A is going to be kind of this ubiquitous kinase that we see for activating a lot of need state pathways. Phosphoprotein phosphatase 1, or also called PP1, is going to be an important enzyme for kind of activating the D-phospho system or turning a lot of these catabolic pathways off. So we're going to spend a couple minutes going through a slide here on phosphoprotein phosphatase 1. Again, this is a glycogen-dependent protein. It's a 2-subunit protein, so it's got a regulatory motif and a catalytic motif. We'll see those on the next slide. And what's kind of unique about it is it's got kind of 3 sort of phosphorylation states because it's got 2 phosphorylation sites. So it can have no phosphates, 1 phosphate, or 2 phosphates. And I promise this is the only time where things get a little bit fuzzy in terms of, well, that sort of doesn't make sense, but we'll kind of put it all together on the next slide here. But again, just to big picture, the function of PP1 is to serve to dephosphorylate things. Remember, phosphatases remove phosphates that kinases put on. So in this case, we're going to consider it in the context of dephosphorylating phosphorylase and putting it into the less active dephosphorylated form. So again, inhibiting glycogen breakdown. As we see at the very end of this mini lecture here, we'll see that it actually serves to dephosphorylate synthase, which actually will activate the glycogen synthesis pathway. So again, 2 subunits, so there's going to be a regulatory subunit, which they call GM for a glycogen binding motif, and then a catalytic subunit. So here is the slide that we'll kind of break apart and talk about more in our in-class activity. But what we can see here are the 2 motifs. There's a catalytic subunit here, which we call PP1C, and then a glycogen binding motif, which kind of is the regulatory subunit, and that's responsible for binding glycogen. So whether this protein is doing its phosphatase chemistry or not depends on whether this phosphatase kind of catalytic domain is separated and able to kind of be removed from that binding motif, or is kind of existing with it. And again, this is where it gets a little bit murky. Now, this part makes total sense. If we are having phosphorylation, we're existing in this kind of phosphorylation state, we would imagine that we're going to be decorating things with phosphates, with kinase activity. So again, we don't want to be having a phosphatase be active. So when we are doubly phosphorylated here, and again that happens with protein kinase A, then we remove this catalytic subunit and it becomes inactive. So and it's easy to get proteins confused here. Don't get this confused with something like pKa, where when we separate the catalytic subunit, that's where it becomes active. Here, separating the catalytic subunit makes it inactive. And this is one of these things that we'll tease apart in our in-class activity, is trying to make sure that we have all these parts working. Now, what gets a little bit confusing here is where we have an insulin-stimulated protein kinase. So normally we don't think about phosphatase activity being associated with a fed state signal. So again, we're going to break this apart a lot more in our in-class activity. But the most active form of this phosphatase, this PP1, is actually when we have 1 phosphate on this 1 position. But again, it's going to be added by a fed state kinase. So again, I promise you, this is the one time we talk about something that's a little bit disconnected with having kinase activity being linked to a fed state signal. We haven't really seen that in the past. Okay, so again, we'll break apart this figure a lot more in our in-class activity, but that should be enough to kind of get you thinking about this. This, again, another figure that we're going to really break down in our in-class activity. Things that we've already spent a good bit of time talking about, which is good, is thinking about this concept of a phosphorylation system and a dephosphorylation system. Again, kinases being responsible for decorating things with phosphates. phosphatases being responsible for taking those phosphates off. So we've already kind of learned about this phosphorylation cascade here. So again, in this chapter, learning much more about phosphorylase kinase. We'll break apart this figure in that regard. But again, since we're dealing with glycogen metabolism, these are really the important players. Whether phosphorylase is active, and again, it's active when it's phosphorylated, or synthase is active, and again, the synthase is going to be most active when it's dephosphorylated. And again, PP1 is responsible for activating this dephosphorylation system. pKa and then its cascade is responsible for activating this phosphorylation system. So we'll spend a lot of time with some guided questions in our activity to walk us through this figure here. So the last thing that we'll spend some time with in our in-class activity is thinking about how some different hormonal signals that we've talked about really play into this glycogen regulation pathway. So two that we've already talked about a good bit are glucagon and insulin. Again, they are work in kind of opposite scenarios. Glucagon released when blood glucose levels drop below 5 millimolar, and insulin released under high blood glucose levels. What's really interesting, again, these are small peptides, again, synthesized and released by the pancreas. So a lot of pancreatic activity that we see with our hormones here. But again, the logic prediction with each of these, everything that we talk about should make sense. If glucagon is released when blood sugar levels drop, we should upregulate glycogen breakdown pathways. If insulin is released when blood sugar levels rise, the logic prediction is that we should upregulate glycogen synthesis pathways. So again, we're going to put that all together with the slide that kind of shows muscle and liver cells, and we'll talk again about some importances of that. Adrenal hormones, again, noradrenaline and adrenaline, also called norepinephrine or epinephrine, differ basically by a methyl group at this position. But for the most part, for what we're going to consider now, they operate similarly. They are activating signals. Again, they activate the fight or the flight stress response pathways. And so, again, we're going to talk about where they bind, and how, again, they stimulate phosphorylation cascades. And again, they're going to be tied to secondary messengers like cyclic AMP and calcium. So again, these four or three hormones really, insulin, glucagon, and then epinephrine, adrenaline, or the norepinephrine, noradrenaline. These extracellular signals are going to be responsible for putting things together in this figure. I'm not going to talk too much about this figure right now. We'll go through it in our in-class activity, but a couple of highlights that are important to mention is we are going to look at how this is different between a muscle cell and a liver cell. So big picture, remember, you have glycogen stores in both your muscle and your liver cells, but they serve different purposes. Within your muscle cell, those glycogen stores are meant to be used locally. So again, one of the things that we'll talk about is whether we have glucose transporters that operate in an influx or an efflux sort of way. And again, you can probably already see here that we have input and output of glucose in the liver cell, but only input in the muscle cell. And that's because muscle cells are only designed to use their glycogen locally. Liver cells, on the other hand, are meant to both use glucogen levels or glucose locally if they need it within their own cells, but they also will export glucose to kind of satisfy a global need for glucose. So again, we'll use this slide, this figure, we'll break it apart in our in-class activity to talk about all the signals that come from the pancreas or the adrenal glands, the different cell receptors that these cells have on their cell surfaces. And then again, once we figure out how all of this works, that'll help us, and we might start our case study. We'll see where we are in time. If not, we'll kind of push that off to another class period to talk about a case study on a patient who has a glycogen storage disease. So again, we'll be able to tie this all together. And then the last thing we'll move on to in our glycogen chapter is talking about gluconeogenesis, how that pathway works, and how that ties into some things that we've already talked about with glycogen metabolism.